Thank you everyone for joining us. My name's Tavia and I'm with Trio, and I'll just run through a little bit of the administration for tonight. So we will be starting with a short presentation on genetics and by Andrea here, and then we will open up the floor for any questions you may have. So you should see at the bottom of your screen a Q&A box. So feel free as the presentation is going on to enter your questions there. And then we will pose them to both of our speakers here on the panel at the end of the presentation. You will not be able to interact, or you'll notice that you're unable to kind of raise your hand. So everything can be typed in into the box, and everything will be anonymous. After tonight, we will be also sending out an email with tonight's session on videos, so you will have access to that as well. And I believe that is all the administration I needed to get through. So without further ado, tonight we have Andrea Jolovic from Genomicare. I almost forgot for a minute there. From Genomicare. She is a certified genetic counselor, and she's actually the founder of Genomicare. So she'll be going through her presentation, as I mentioned before. And then from Trio, we have Dr. Arielle Cantor, and she is a reproductive and endocrinology and infertility specialist here at Trio. And she practices in all subspecialties related to the fertility field, with a little bit of a keener interest on the genetic side and looking at genetic testing. So that's why we asked her to join us tonight. So without further ado, Andrea, why don't you start us off?
Great. Thank you so much for the introduction. And like mentioned, I have a slide deck that I will review. This will be available to you. You know what? I'll share it as a PDF too if anybody wants a PDF copy for the records. But our main overview for tonight, sorry. Oh, and actually, before I move slides, this is our phone number and an email for you to get in touch with me directly if you need to, if you have any questions kind of outside of this presentation. But going on, I'll have two real main goals today. And both of the tests that I talk about are available to everyone. Okay. The first test is a review of pre-implantation genetic testing. And the second test is called expanded carrier screening. So PGT-A, it's a genetic test on the embryos. Expanded carrier screening is more of a test in parents. So we'll get started. Starting with PGT-A first, pre-implantation genetic testing for aneuploidy, or sometimes, like I said, referred to as genetic testing on the embryo. Let's go through the acronym really quickly. So the P in pre-implantation just means that the embryos can get tested before they are transferred and implanted back into the uterus. G is the term for genetic. This is where the test focuses on really, honestly, really high level genetics of the embryo biopsy, specifically looking at the chromosomes and the DNA in the embryo. We'll get into more of what that means in a slide or two. The term T is for testing as well. More on this later. The only term I really want you to remember after our call today is the term A for aneuploidy. This is the scientific term for chromosome imbalance. So as soon as we have too much genetic material or not enough genetic material, we call that aneuploid. So let's go back to high school biology for one moment. If you, for example, took a pinch of skin and you looked at the skin cells under a microscope, and then you zoomed in even further and just looked at one skin cell under the microscope, what you would see in the very center is these X-looking structures called chromosomes. And all one chromosome is really, really tightly packed DNA, and DNA is what carries our genes. So we've got our hair color, eye color, how tall we are, everything about us is coded by sets of our genes. And then our genes are found on our DNA. And then if you take that really long strand of DNA and you squish it back into its smallest container, that container is called a chromosome. The reason I say all that information is because as humans, we have a lot of genetic material. Specifically, we have 46 chromosomes, those containers, we have 46 total that come in 23 pairs. And in each pair of our chromosomes, we inherited one copy from our dad or the sperm. We inherited the other copy from the egg or our mom, and so on and so forth through all of the pairs of chromosomes. So what we're looking for when we do PGT-A is looking to see, is the embryo predicted to have this complete set, all of the genetic material, or not? Now, I will just flag that as humans, we have these 23 pairs of chromosomes, numbers 1 to 22. We all have, men and women. It's the last pair here that are called the sex chromosomes that are if we have an X and a Y, that's typically for male. And then imagine the Y wasn't here on the slide and there was just two X's side by side. That's typically for female. Okay. So XX female, typically XY, typically male. Now, at the time of biopsy, we can see all of the genetic material, all of the DNA, really. We can read all of the DNA, but we do not disclose the sex of the embryo in Canada, typically. There's definitely exceptions to this, but we do not see this data. I don't get that. Your doctor does not get that. Okay. Taking one step back for the overview of where does genetic testing on the embryos fit into the IVF cycle. So what we have in the very first steps 1 to 4 is just IVF. We have hormone therapy, ovarian stimulation as directed by your physicians and your nursing staff. Then you have an egg retrieval and sperm collection, usually on the same day. And from there, what happens is all of the mature eggs that were retrieved are fertilized with sperm. And those that are successfully fertilized, we watch as they grow and develop to about day 5, day 6 of embryo development, sometimes day 7. And it's at this stage of embryo development that we can take a really small biopsy of cells, send those cells to a genetic testing lab to see what are the results of this embryo. Do the cells look good? Does the genetic material look good? Or is there something detected that would reduce the risk of that embryo working? Reduce the chance of that embryo working. That biopsy goes to a lab in the States. It's called Genetics, while your embryo stays at Trio, safe. And we basically wait for the results to come back to decide which embryo to transfer first. Now, looking a little bit at the embryo. When the embryo reaches day 5 or 6 of development, it is something called the blastocyst, okay? And it is made up of 2 different cell lines. So the first is this cluster of cells here. It's called the inner cell mass, and this is what develops into a baby in an ongoing pregnancy. And then there's this outer layer here called the trophectoderm, and the trophectoderm is what develops into the placenta and other tissues that support an ongoing pregnancy. When the embryo is day 5 or 6 days old, this is when the embryology lab at Trio can take a really small biopsy of about 5 to 10 cells. And these are the cells that are sent to Genetics for testing. And results typically take in the range of 3 to 4 weeks. Okay, but team can confirm. When the biopsy is sent to Juno, the results can come back one of 3 ways, 4 ways, one of 4 ways, but these are the main 2. The first is a result called negative for aneuploidy, or also known as Euploid in some online literature. This means that there is predicted to be a correct number of chromosomes per cell in the biopsy. So we have that 46 total, there are 23 pairs, everything looks good, and these would be the ones that are prioritized for transfer. The other result is something called positive for aneuploidy or aneuploid. This means that there is an incorrect number of cells predicted in the biopsy. And, there could be more chromosomes or less. And we would predict that embryo would have a very low implantation rate or a very low ongoing pregnancy rate. And we would basically put it at the bottom of the list, and we would not recommend those embryos for transfer. More rarely, there is a result called a no result or an inconclusive. It occurs in about 2% of all embryos that undergo PGT-A. And basically, what it means is there was a biopsy that was taken, it was sent off for genetic testing, and for many different possible reasons, the lab is not able to make a call on the result, not able to make a call on the embryo biopsy. So it's like you just did IVF and you don't know the genetic makeup of that biopsy. There are a bunch of different reasons that a no result can happen. Sometimes it's just because the embryo quality itself, the embryo is of poorer quality and it's difficult to visualize. So when the embryology team took a biopsy, the cells didn't contain DNA. It could be that, again, there wasn't enough DNA collected at biopsy. everything could have worked perfectly with the biopsy, but the DNA could degrade prior to getting it tested, or everything worked perfectly, but then at the very last stage, the DNA profile, the graph readout that the lab does is unclear. Often the reason why somebody gets an inconclusive result or a no result is actually unclear. Like we can't give you a reason usually, but typically it can be considered for transfer, or it can be an embryo that is rebiopsied after you meet with your team. So just highlighting the three results. Euploid or negative for aneuploidy. Like I mentioned earlier, it's the highest priority for transfer, these embryos. If there's more than one embryo that is considered euploid, you would consider the embryo grading, or your team at Trio would consider the embryo grading to determine the priority of transfer. Then, of course, like I mentioned, there's this no result. It can be absolutely considered for transfer. You can consider it for rebiopsy. And we would give you the age of aneuploidy. So the chance that that embryo is chromosomally abnormal, we would quote based on maternal age. And of course, just further discussion can happen around this embryo. And then looking at the aneuploid, the one also known as positive for aneuploidy at Juno Lab, it's not typically transferred, although there can be exceptions. And the breakdown within aneuploidy, sometimes there is further discussion recommended for certain results. I like if there's a result like a segmental aneuploid, that warrants a little bit of a bigger conversation sometimes. Now, looking at PGT-A accuracy and limitations. So we say, regardless of age, all couples are at risk of producing an aneuploid embryo, making PGT-A an option for all. Juno Genetics quotes an accuracy of 98%. So what this means is that they can technically call an embryo good to go, euploid, but it's actually abnormal aneuploid. The chance that that happens is low. It's about just over 1%. And the inverse is also true. It could be that they call an embryo abnormal, aneuploid, but it's actually good. That's even lower risk, lower than 1% typically quoted. So it's just me saying that this is not a diagnostic test on your embryos. It's a screening test with the goal to help you prioritize which embryo do we transfer first, which embryo do we transfer second. And really the goal is to get you to pregnancy sooner by not unintentionally transferring an embryo that was never going to work. So it's just me saying it's not perfect, but it can provide a lot of helpful information and like I said, get to pregnancy sooner is the goal. Now, a really big limitation or a big thing for you to be aware of is PGT-A doesn't look for everything. It doesn't look for family history of anything. It doesn't look for, autism spectrum disorder, cancer, anything like that. It is just looking at does the biopsy have the right number of chromosomes? And therefore we can predict, we can assume that the rest of the embryo is good as well, or not. That is it. If there is a family history of something, a genetic condition already in the family that you're aware of or something you're wondering about, there are other types of tests, but PGT-A would not look for anything specific. Now, a euploid embryo, it doesn't always implant and lead to an ongoing pregnancy. And this can be really frustrating. After doing an entire cycle of IVF with PGT-A and transferring an embryo that still didn't work can be really frustrating. Today, we're quoting that a euploid embryo, so an embryo that chromosomally everything looks good, the chance that it implants is up to 65%. Sorry, the chance that it implants and leads to an ongoing pregnancy is 65%. Now, it is possible that after you do a cycle of PGT-A, that there isn't a good embryo that we would recommend for transfer. And this is another really frustrating part with PGT-A. But after conversation with your doctors and nurses, this can be something that you can understand a little bit better if you're at risk for this outcome. Now, regardless of doing PGT-A, there remains a 4 to 6% background risk for birth defects, congenital anomalies, things like congenital heart defect, the way the brain develops, different organs, things like that. This risk exists regardless, and this could be identified during a pregnancy or after birth. Now, when our team says that all couples are at risk of producing aneuploid embryos. It comes from data that shows that as maternal age increases, the chance of aneuploidy also increases. This is because women are born with all of their eggs, and as women age, their eggs age with them. So with older age, all it means, and I shouldn't say with older age, with across all ages, there is a chance that an egg doesn't prepare its genetic material correctly right before it's fertilized. And when we, this slide is really maternal age mostly because we know that when we look at where does the aneuploidy originate, where does the chromosomal difference originate from in an embryo, typically 90% of the time, 90 to, depends on the study, but about 90% of the time it is of maternal origin. So that means that sperm can absolutely contribute chromosomal aneuploidy. It's harder for a sperm to contribute a big chunk of missing or extra genetic material because if a sperm is missing too much or has too much extra, they typically just don't survive or they don't look good. They're not formed properly. So again, just leaning towards the biology of where aneuploidy comes from is maternal most of the time. Regarding age, I'll leave the slide up for you just to see if you can see where your age is. But we see an increase in aneuploidy really from 37 onwards, 36 onwards, kind of 37 onwards really is usually where we see an increase in aneuploidy. And a little bit different to genetics and genetics and fertility, but I did want to talk about something called IVF attrition and just for you to be aware of timelines. So when we talk about IVF, every step of the way we can see a little decrease in the numbers that we have. So, when you have an egg retrieval, if you are doing IVF, when you have an egg retrieval, you may start off with a number and that number might decrease right away. Typically, I'm going to quote 80% of all eggs that are retrieved are mature, but I'll let Dr. Cantor mention if any of that is different at Trio. From there, we expect 50 to 70% of all mature eggs to be fertilized. And after that, as we watch the embryos grow and develop, we do expect 30% to 50% of all fertilized eggs to grow to day 5 or 6. After that, if you consider your age-related aneuploidy, which if you're, under 35 is maybe 1 in 3. So, if you have 3 embryos, maybe 1 could be aneuploid. If you have 6 embryos, maybe 2. Now, I am absolutely using population data on like a single cycle of really, a small number of embryos, but it's just for you to be aware of, okay? And then, like I said, results are typically back 3 to 4 weeks after your biopsy. Now, that is the conversation on genetic testing on the embryos. I want to switch gears a little bit and talk about genetic testing in parents. It's called expanded carrier screening, and it's a test that is recommended before you proceed with IVF and PGT-A. Also, if you're doing IUI, it is absolutely an option, actually, prior to conception of any type. So, what is it? It is carrier screening is a genetic test that is looking to see if to identify couples who are at risk of passing on a serious genetic condition to the next generation. It is performed on an individual. Typically, you have no signs or symptoms of carrier status. It really is just something that we all carry. We all carry these different genetic conditions. We have no signs of it. We have no symptoms of it. We usually just find out that we're carriers if somebody in the family has a child affected with the condition, or if we do a test like this. Like I said, the goal of carrier screening is to provide future parents with information, basically to guide pregnancy and future planning. And we say that today it's estimated that about 2 to 3% of couples are at risk of being carriers for the same condition. So a lot of people do this test and it's really crossing their T's, dotting their I's to make sure that they're not at risk. But for the people who are the 2 to 3%, it is absolutely life-changing. So what the test is, it's big. It's looking at a lot of different genetic conditions that are pediatric onset and typically severe and passed on in a form that is known as recessive inheritance. So one more time, back to biology class, high school class. 2 people both have to be carriers of the exact same condition, like these in this picture here, in order for a child to in order for a child to inherit no working copies of the gene and therefore be affected by the condition. So in this situation, when two people are both carriers of the same condition, they have a 1 in 4 or 25% chance of having a child affected with whatever that condition is. And some of the really common conditions are things like cystic fibrosis. There's about a 1 in 25 carrier. 1 in 25 people carry this condition in the population. Spinal muscular atrophy, just the way that the muscles develop after birth, or I guess the way that the muscles work after birth, is impacted by this condition, and about 1 in 35 people are carriers. So some of these conditions are actually pretty common in the population, and then they do get more rare as the panel has gotten bigger. But I want you to know that within this test, about half of the genes on the panel are associated with a shortened life expectancy. 25% of the conditions, about a quarter, are associated with limited or no treatment options. So they are severe without many medical management options available. About a quarter of the conditions are associated with a risk for intellectual disability, and then about a quarter of them are associated that they do actually have treatment options. That if we know about them early on, it can improve quality of life. These don't add up to 100%, we're aware. It's just there's a lot of overlap in the way that the conditions present themselves. So it's just to give you an overview of what they are. I won't go into the slide in too much detail. I just want you to know that the reason we offer this is because, especially at the preconception stage, it allows for families to maybe add on additional testing in their IVF cycle, maybe consider donor gametes, maybe explore the option of adoption. And at Trio, the third-party program is already very familiar with expanded carrier screening. All sperm and egg donor, I think internationally, all sperm and egg donor banks already test their donor pool. So all sperm donors, all egg donors have genetic testing results available. So we already know what their carrier is for. So we're with the third party program, we're testing to identify what the intended parents are carriers for, just to kind of, again, cross T's, dot I's, make sure that everything looks good before IUI or IVF. Of course, there are options during pregnancy as well. I won't get into those. And then after birth, management and therapy, absolutely. If you're aware of the condition before birth, we can make a plan for management. When I say these tests are big, they're really big. We're looking at up to 613 different genes that's associated with more than 1,000 different conditions. So it's a pretty big test. And then I think if you're interested, usually this test is 613 is good. If you're interested in more, this lab just launched a bigger panel. It's 835 genes. It's I say 613 is big, 835 is really big. Results take 3 to 4 weeks. If you're interested, you can let me know. If you're just interested in talking about it, you can let me know. You don't have to, want to do the test. I am wrapping up here, but I wanted you to know that for the Q&A, please go ahead and ask any questions. But if you have a personal question, or if you have like a question about your family history or medical history or something like that you want explored, you can actually book an appointment with our team. There's a 15-minute intro call with a genetic counselor. There's no fee associated with it. Usually the appointments end up being with me, actually. Or if you're interested in expanded carrier screening, one of my colleagues, her name is Vernie, she's amazing. You can set up a 15-minute intro call just talking about expanded carrier screening if you want. Or you can just email. We have a form here. Okay, I'll stop sharing.
Great, thank you. There are some questions here already in our question box. And the first question is, what are the other tests that you were speaking of for autism and cancer and things like that?
Yeah, that's a really good question. So when somebody presents to our team or any genetics team with a family history, there are often diagnostic tests that are available to families. So what we offer, or what we recommend, is if you have a strong family history of cancer, we always say the best person to have genetic testing is somebody who has had that diagnosis of cancer, to see if we can identify a genetic cause of the cancer they have. That isn't always possible, we're aware. So there are teams like ours that offer proactive cancer panels. So you can look at like a bunch of genetic causes of cancer in a person that's unaffected, to see if they carry something. Again, it's not perfect, but it's a really good option when an affected relative is no longer available for testing. With autism, same thing. Or autism, I shouldn't say autism. Autism plus. So we can offer genetic testing for individuals who have a history of autism plus seizures plus developmental delay, intellectual disability, that kind of thing. So if it's just, having family members on the spectrum and that's it, everything else, they have no other health concerns. Usually we say that's multifactorial, a little bit of genetics, small genetics added up, and then plus environment for presentation. But if it's a more severe presentation, those people are usually eligible for genetic testing. If they have something identified, then we can talk about offering their family members like you tests at the IVF and PGT stage.
And I believe this is tied into the same thing, but what if they want to test embryos for family genetic conditions?
Yeah, really good question. So we would need to have the family genetics result. I would need to have a genetics answer already to be able to test the embryo. So PGT-A, it sounds really high tech and amazing, but we can't tell a lab, go test this embryo for cancer. We have to tell the lab, this family has this genetic cause, and they have to be they have to basically look at that very specific spot in the gene. It's like zooming in on Google Maps into one little tiny tree in a backyard to look at that tree. Does that tree have all its branches? So it's very, very specific. We can't tell them to just do an overview of cancer genes.
So, like you said, having the answer key is necessary to kind of overlay over top of that
Hmm.
information.
Hmm. And we can help families, by the way, too. Like we can help families try to identify genetic causes before they even get to the IVF PGT-A stage, but that does take time.
Another question, if all embryos are considered aneuploids, can you still request one to be transferred? This is probably a Dr. Cantor question,
Hmm.
at Trio in the hopes that it's part of the 1% that might have been miscategorized.
Hmm. Yeah, so that's a good question. So I will say, typically, if I had a patient whereby, their whole embryo cohort was genetically abnormal, that's always a really difficult position to be in. And obviously, that's not a cycle outcome that we're hoping for. Typically, in those cases, I have had some requests of patients, ask, is it possible that, we could potentially still consider a transfer as opposed to potentially, undergoing another stimulation and trying to make another cohort of embryos, because that's, of course, a big venture to go through. Typically, what I would do is I would recommend that that person would meet with genetics to discuss the specific aneuploidies at play. So, there could be, differences in the chromosomes that are affected. So, typically in that case, I would definitely have them meet with genetics. After meeting with genetics and being as informed as possible, it is a discussion to be had. So it's definitely not black or white. It's not, full out no, but I think it's just very important for the person that's considering transfer of an embryo that, potentially could, lead to a viable pregnancy, really understand what the possible risks are. I will say that, and correct me, Andrea, if I'm wrong, but a lot of these aneuploidies, typically wouldn't even result in a live birth. But there are some that may. So I think as long as the person involved is as informed as possible, I think it's really a joint decision between the patient and their provider. I know that there are some of my colleagues that may not feel comfortable doing that, that type of treatment, but it's definitely something that's come up before in practice, and it's a discussion to be had, and we involve genetics in those conversations.
I will just add one thing that I just noticed from the question. It says that the 1%, or in the hopes that the 1% that may have been miscategorized. So Juno Lab with a different clinic, I think a clinic in New York or something, they did another study, a separate study where they looked at how many how many embryos that were aneuploid implanted and how many led to an ongoing pregnancy, and they actually found 0%. So they found actually not even that one, there wasn't even a percentage that implanted. When Juno called an embryo aneuploid, 0% implanted, there was like 100 embryos they transferred. 0% implanted, 0% led to an ongoing pregnancy. So that would be the data we often share. Yeah.
Right. Thank you for sharing that. Um, if an embryo doesn't make it to the blastocyst stage, or if it does, but then miscarries early on after transfer, does that mean they were likely aneuploid? Aneuploids? I think this is probably a
The
longer than just this discussion, but
Okay, you know what? Let me try really quick. Usually we say that if a euploid embryo doesn't implant or leads to, a miscarriage, but uterine environment, and Dr. Cantor, please, jump in too. But uterine environment, everything looks good. Of course, morphology looked good. Euploid embryo looked good. We say that it's possible that it was still something genetic, but something genetic and so small that PGT-A can't detect it. Like we're talking single gene things that like cannot be seen at the lab level right now. So we suspect that that's probably more likely. So
I think that
not that it was aneuploidy is what I was trying to say. Yeah.
right.
But I think just to touch on the question further, I think if an embryo isn't even making it to that stage of embryo development, most likely it's because there is some, egg or sperm quality issue that's not allowing for embryo progression. So typically if, to reach the blastocyst stage is usually when an embryo has reached a specific stage of embryo development, usually around day 5 to 7, typically. But if it's not even making it to that stage of development to be able to even be tested through PGT, which can, sometimes happen, that's most likely meaning that there is some egg or sperm quality issue that's not allowing for that embryo growth pattern.
Right. The next question is about how much is extended genetics testing? I'm assuming this is the panel that you were talking about, not PGT-A. So I don't know if you know that.
Yeah, the price per person is $675 through our team. The like smaller one, the one that I think is more than good enough. The bigger one is I think 750.
And who should consider getting expanded carrier screening?
So there is a policy or a paper that came out in April 2025 by the Canadian Association of Gynecologists and Genetics, did a joint paper. So they say that everyone preconception should get screened for, off the top of my head, I think it's like 10 different conditions. Okay. All 10 are covered in that 613 that I mentioned. The problem with their recommendation, and they state it in their recommendation, is that as a country, we cannot fund it for everyone, and we don't have the capacity to run it for everyone. So they're basically like, everyone should get it done, regardless of ancestry, regardless of ethnicity, regardless of family history. But we just can't, like, as a country, support it. And then that's it. That's like they finish it like that. So I'm biased, and I say that everyone prior to pregnancy can do it. And if you're a heterosexual couple, starting the female is totally fine. We test the female, we review the results, and then if you're like, oh, we're happy with that, you don't even have to sometimes test the male. Sometimes we do everyone together, same time. So there's options. So if you're even slightly interested, just let us know. It's like there's no fee to meet with us. And you can kind of ask all of your specific questions. We can definitely help.
I'll just add a little bit. I think, once again, it's a consideration that anybody can do. But there are certain subgroups that you might consider it maybe a little bit more strongly. So, for example, I do have some, couples that I see that are like have a familial relationship. So, they're first cousins, or, they have some sort of, you know, they're related in some way, like they are potentially more at risk of having a genetic condition that's in common between the two of them. So that's something that I discuss with them, I emphasize a little bit more strongly in those cases, because I have picked up, commonalities that might not be, as potentially expected in, people that are otherwise unrelated. So that's one subgroup whereby, it's something that I definitely bring up with. It's also something that, we didn't really get into it too much, but with our third party program as well, we anyone who's considering donor gametes, either donor sperm or donor egg, it's something that we typically also, it's part of our policy just to make sure that there's that genetic compatibility before going forward with a pregnancy. But once again, it's something that is available to anyone who's interested as well to discuss further. It's also something that a lot of people that especially have been in the US, it's something that's, definitely a little bit more emphasized than is here. But it's something that we offer and is available to anyone who's interested.
What is the embryo grading process, and is it an additional fee from the PGT-A? I guess, Dr. Cantor, if you want to touch on this,
Yeah.
Trio.
Yeah, so embryo grading is basically a morphologic assessment of the embryo development. It's something that's done by our embryologist. It's part of the IVF process. There's no additional cost to that. Basically, once the egg and sperm come together and we grow the embryos, they're assessed at different points in time. And typically, when we're grading an embryo, most likely you'll notice that there's a number and letter scheme by which we explain how the embryo looks. Different clinics do have different descriptions, so you might, depending on where you've been, you might notice some differences. But at least at our clinic, our embryos are typically morphologically graded from a number scale of 1 to 6, and then there's two letters associated with the grading as well that really describe how the inside of the embryo looks and how the outside of the embryo looks. And it's basically, ultimately, it's a description of how developed the embryo is based on its morphology. It is important to know, however, that morphology doesn't necessarily dictate genetics. So you can have a very nice morphologically looking embryo, and that doesn't necessarily guarantee that it's genetically normal. Also, vice versa, you can have an embryo that, might not be the prettiest, but it could be normal. So, I have some couples that ask me, well, my embryos look nice morphologically. Is PGT necessary? And they're two very different processes. So the way it looks doesn't necessarily dictate the genetics. So it's important to kind of appreciate that piece as well. But, if you do PGT-A, then, let's say you had a group of genetically normal embryos, and then we're deciding which one to put in first. There's a variety of different, factors that the embryologist will look at, and the way the embryo looks is one of them.
Great. And I see there's a few questions for people who joined us late. So I just wanted to let everyone know we will be sharing this presentation. It is being recorded. And as soon as we have it online, which should be later tomorrow, we will send an email out to everyone who RSVP'd to our talk here. So we will get that to everyone. And thank you for explaining that for us there, Dr. Cantor. The next question, if I only have one frozen embryo, would you recommend doing PGT-A on it?
So I think it's really a personal choice. Truly with my patients, I talk about PGT-A with everybody. It's something that, regardless of age, it's a consideration, and there's different rationales to go about it. There is cost associated with the test, but there is, value in the information if it's something that you're interested in knowing preconception. I think if it's information that you really want to know upfront, even if there's only one embryo, I think there's value in that information. I do have some couples whereby, if they only have the one, they want to put it in and see what happens. And I think that's also very reasonable. So at the end of the day, I think it's, a discussion that I have with everybody. And then, it's something that we can reevaluate. It's also sometimes hard going into a cycle with, I always try to set expectation in terms of how many embryos we may expect, but sometimes, it doesn't always turn out that way, or numbers are a little bit different. There is the opportunity of, let's say you want to plan for PGT, but then if there's more attrition than anticipated, sometimes you can, opt out or change your mind. So, but that being said, I mean, I think even if there's, not many embryos to test, I mean, I think there's still value if that's information that you want to have going into an embryo transfer potentially.
If there are 10 day 5 blasts, I guess either, I'm not sure if they're fresh right now or frozen, but can you test 5 now and test 5 later just to help with the costs?
So definitely. So typically in terms of the cost of the PGT, there's usually a fee to biopsy the embryo cohort, and then there's a fee to test each embryo. So the more embryos that you make that you potentially want to test, the more costly it can become. So typically, usually before biopsied cells are sent off, we confirm of your embryo cohort, how many do you actually want to send off to test? Some people will want to send off, their whole cohort, and some people will only select a subset because, it can become costly. Typically, the whole cohort is biopsied, but only cells of the embryos that you request are sent off. So hypothetically, let's say you didn't conceive with those embryos that you did test, or if you wanted to, send off those additional cells in the future, it can be done. But most definitely, you don't necessarily have to test your whole cohort if you are lucky enough to make that many embryos.
Someone is asking about the cost of the test, which I believe has mostly the panel has been the panel one was answered. And then, as Dr. Cantor was referring to at Trio, there is a fee for the biopsy, which is $2,350. And then there's a fee per embryo for testing. And I believe it's $425, I think. Yeah. Per embryo. So those are the two different costing structures depending on the test you're talking about. Is there a reason why a couple would only keep producing aneuploid embryos? Is there a type of testing that can be done to select a specific egg or sperm that's likely to turn into euploid embryo instead of only testing at the embryo stage? Oh, I guess they're asking if there's an earlier stage other than embryo stage that tests can be done.
Yeah, I mean, I think this is probably a question we can both answer a little bit. I mean, I think these are one of the hardest things to deal with when you have, people going through IVF, and unfortunately you're making embryos, and unfortunately none of those embryos are usable. I think often, it really boils down to, egg quality, sperm quality, and age is the biggest factor. I often see these cases in someone who's going through IVF and they're older, and even if their, ovarian reserve or egg number is, higher than expected for their age, unfortunately, we can't change the inherent genetics of our eggs and our sperm. I wish we had something that I can give or change to, change that inherent nature, but we don't really have anything to change the eggs that we retrieve from the cohort that's being stimulated, or, even if, when we're doing IVF, and we're selecting sperm, the embryologist typically picks a sperm that morphologically looks appropriate. Once again, morphology doesn't necessarily dictate genetics. So, we don't have something that can change, the genetics of our embryos, unfortunately. And that being said, I've had people go through IVF more than once, and we've had, one or two cycles where we have no usable embryos, and we've had, a subsequent cycle where there are embryos that are usable. So, it doesn't, I know IVF is a big process to go through physically, emotionally, financially, etc. So, sometimes it's, not always a possibility to go through more than one cycle. But, there are there are times where, not every embryo that you make, depending on, the cohort that you're recruiting, sometimes you can have a bit of a difference there. But typically this boils down to egg and sperm quality.
I wanted to say the same thing. Our team has absolutely seen two cycles, same couple, same clinic, same doc. Everything is the same. The only difference is the cycle, and the outcomes are literally sometimes black and white. So I think a lot of times we tell families, if you haven't been told by your doctor, the reason for having more aneuploid than we would expect for you is probably truly just the outcome of that cycle. It doesn't define, what you and your partner can produce. Doesn't define kind of what a future cycle would look like if you haven't been told otherwise. So yeah, we've seen the same thing. The only time where we may see more aneuploidy is sometimes people carry genetic translocations. What that means is, like, I showed you that picture where, we have two copies of all of our chromosomes, and that's kind of great. Some people have two copies, like two chromosomes stuck together. For example, they still have all the genetic material they need. They have no health concerns with that. But when they go on to make egg or when they go on to make sperm, they can pass on too much genetic material more often, or not enough genetic material more often. And so we do see those families producing more aneuploidy than we would expect. But I think actually Juno Lab is actually really good at kind of flagging those families. So yeah.
Another technical question here. If eggs retrieved all had central granulation, could this mean it is aneuploidy, even if it was determined to be euploid? You might want to read that question over if it's something that can be answered here, or if it's too specific.
So sometimes the embryologist will make certain comments in terms of what the eggs look like at time of egg retrieval. There are certain features to the eggs, if they look more granular or if they're darker or, certain descriptions of the fluid in the eggs as well can sort of point to a poor egg quality picture. That being said, I've had cases where, there's a lot of comments from the embryologist, and it doesn't necessarily always dictate, the genetics of the embryo itself. So I do, take those comments and it doesn't always dictate ultimately
Right.
embryo genetics. So there is some meaning to it. It could kind of give some sort of description of what the egg quality is. Um, that being said, if those eggs had those just that depiction and the embryo ended up being aneuploid, um, I would go along with the PGT result. I wouldn't, say that there was something, I wouldn't say it's actually euploid or whatnot, but I think the embryologists do give us certain, morphologic descriptors, especially in early egg and sperm before they reach that blastocyst stage to, give us some explanation. So for example, let's say you don't even reach a blastocyst, that can kind of point to some reasons as to why, based on what those eggs and sperm look like before they even made it to that stage, that could have been PGT'd.
If while we're on this topic, I just scrolled down because I thought I saw another question on the same lines of this coming from the probably comments that happen in the lab and the gradings. How do you counsel patients, or how does Trio counsel patients on if they're only sending a certain amount in the cohort out? Which ones should they send out for testing? Is that based on the embryo grading?
Yeah, so good question. So, if you're deciding, from your cohort, if you're only going to select a few to send out, typically we would choose or recommend to choose, your embryos that are morphologically graded the highest and have reached their developmental stage, in the expected time frame. That being said, I will say implantation potential of day 5 and day 6 embryos is similar. And also, as I mentioned, morphology doesn't necessarily dictate genetics. So, you know, it really depends, I would say, on the clinical case. I would say it depends on, your age, how many embryos you have, what the embryos look like, but we can help guide if you want to, select a subset of your embryos to send off and you're not really sure how to go about doing that, we can kind of guide, if you decide you only want to send a certain number, how to go about choosing your best cohort. We have tools beyond morphology as well. And genetics. We have, internal scoring systems in the lab if we're using our special incubator called an embryoscope that gives some other grading to our embryos as well. So there's a variety of ways to kind of assess, which embryos to select for, which we would choose for potential transfer.
And a question over to Andrea, talking a little bit about your role as a genetic counselor and what you think might be least understood by patients or even by other providers.
That's a good question. I think the biggest part is that genetic counseling does not mean genetic testing. So if you're wondering about whether you want to access genetic testing or whether genetic testing is a good option for you and your family, you can meet with a genetic counselor. So biggest preconception or like misconception, sorry, is that meeting with a genetic counselor means you're definitely getting the test. Don't worry about that. We chat with you and kind of help you decide if it's the right thing.
It looks like there's a few questions, I think probably on Mosaic,
Mm.
which I know is a whole different topic because of Juno, but let me read this one out. Based off your clinical experience, what do you typically see for a transfer of an embryo with inconclusive PGT-A? I'm assuming they're referring to a Mosaic result there. And then there was another specific question about Mosaic results.
Dr. Cantor, do you want to chat about the inconclusive more and then I'll
For inconclusive, are we referring to no result embryos?
right, yeah.
Oh, okay.
Yeah.
Okay, sorry.
So if ever I have a patient that has an embryo that's of no results, we discuss, options. So One option is that the embryo can be thawed and rebiopsied, and we can, attempt to get an assessment of that embryo again, and that's done without added cost. So if you had an embryo that was of no result after PGT testing and you still, wanted to pursue trying to get, that PGT result on that embryo, we could retest it. It does subject the embryo to an additional thaw refreeze. That being said, typically the risk of the embryo is still relatively low. So, I have a lot of patients that I see that we consider, thawing and rebiopsying. That being said, if we have a large cohort of embryos and we have a few euploids in there with a no result, we might say, you know what, let's sit on this, we'll keep it on the side, and at some point we can consider rebiopsying it if needed. We might not even need that embryo, so it might not be our first go-to. Alternatively, we could also treat it as just an unknown embryo. So, even though it was biopsied, we don't have, for whatever reason, there wasn't enough DNA that was amplified to give us an answer of if this embryo is genetically normal or not. So, we can also just say, we don't know what this embryo is. We can just, assume it may be normal, it might not, and just aside, if we don't want to, subject it to an additional thaw refreeze, we can just put it in and see what happens. So, we could treat it as almost, an untested embryo in a sense. So ultimately, it really just, it's something that I talk about if we have those embryos that occur. It doesn't happen, in every cycle or, very often, but when we do have them, I talk about, do we want to, rebiopsy and reassess, or do we want to just put it in? Sometimes we're fortunate that we can just keep it on the side and we might not even necessarily need that embryo and it's not, at the forefront. I do have some couples where it might be our only embryo or all the other embryos are genetically abnormal, and then it becomes a bigger discussion. But it's definitely something that we talk about and there's options for those embryos.
Um, all right, there are a few different questions about the risks in close familial ties, so cousin marriages, and kind of the risks around that. So I wonder if you want to touch on that.
So we quote a 2 to 3% risk for couples. 2 to 3% of all couples are at risk of carrying the same condition, okay, in the general population. When people share a common ancestor, so cousin marriages, or sometimes, actually, I'll say cousin marriages, the risk is double. So it goes from anywhere from 4% to 6%, okay, chance of being carriers of the same genetic condition. Carrier screening looks at the panel I mentioned, 613. Maybe for families that have shared ancestry, I would recommend the bigger panel, but even the bigger panel is 835 genes, right? We have as humans 20,000 genes. So when we do the testing we can do today, even if when it all comes back normal, we can't rule out that there is something super rare that two people both carry. And it's really just because I actually never said this. We are all carriers of something. Every single one of us on this call carry actually multiple things. We just don't know what that is that we carry until we do testing or until, like I said, it happens in the family. When two people have common ancestry, they share, they are related by blood. The chance that they carry the same thing is obviously a lot higher. So we can't rule, even with a negative genetic test, we can't rule it out. But a genetic test is a really good start to see, like, is there something more common, manageable, treatable that is in the family that we can then, control for, or not even control for. You're aware, and then we chat about your options. I think I answered that.
Yeah. um, at Trio, do the doctors discuss carrier screening with the patients, Dr. Cantor, or is this done before by the patients on their own? How does that process work?
So it's a good question. It really depends. I have some people that come to me and they've already done some form of screening already. I have some people that have, done 23andMe or, some other, other testing that they want to, expand on further. And it's also something that comes up in discussions as well. So, I have some couples that, or individuals that want to do additional screening. It's definitely something that we talk about, and it's something that we can help arrange for you as well. As I alluded to before, it's something that it's part of our third party program. So anyone who's pursuing pregnancy with a donor, it's definitely mandated. Um, there's certain scenarios where, once again, I bring it up a little bit more heavily, but, it's something that can be offered to anybody who's interested. So, either the provider will bring it up, you can bring it up. Maybe it's already come up prior to even coming to the fertility clinic, and we can expand on it further.
There is a specific question, a few questions specifically about third-party reproduction. Is there anything to expand on regarding genetic carrier screening for third-party reproduction in particular?
I think the biggest thing is when you're picking a donor, as Andrea mentioned, I mean, we all carry something, right? And, typically being a carrier for something, you're completely asymptomatic. You would have no idea that you're a carrier for that. So you really don't know unless you test. I think the biggest thing is, before you even, doing the genetic carrier screening kind of really helps narrow down your donor selection process to really help guide you in terms of picking a donor that you're compatible with in a sense genetically. It really helps you select your donor because you basically just want to kind of screen, and choose someone who doesn't carry something that you potentially do have. So it really just, helps you in that way. It's a way to, among many ways of, screening and selecting a donor, that's an additional thing that, one would consider when they're picking a donor before doing
Right.
so.
So people are doing that before they even get
So,
into
so
selecting
typically,
their donor.
yeah, so typically it's part of the, process that's involved in, leading up to donor selection. You would do your expanded carrier screening, and then Andrea, you can touch on it a little bit as well. But, typically once, the carrier screening is completed, then, you would meet with our, genetic counseling team and, have consultation in terms of making sure that, you and possible donors are compatible.
Yeah, exactly. So you would meet with our team before purchasing, and we review your results, compare them with the results of. Usually, we actually ask for 2 to 3 donors, unless you are, you really have chosen down to one already. We usually say, hey, you pick your top 2 to 3, and we review it with you. We go over all the conditions you carry in a lot of detail. We go over all the conditions that the donors carry with a lot of detail. And most of the time, we just say, you're good to you're clear, good to go, from our perspective, and then you purchase from there.
And we have a few more minutes here. I do notice that there are many questions on the on embryo grading, and that is something we know there's great interest in. And we are going to have our lab director or one of our senior embryologists do a talk on this. So I will let everyone know once we are doing that. I believe we're going to do it in the next month or so, because there is a great interest in our trio community for that. So for the last few, and then also I wanted to also point out, especially for the people who came a little bit later, we will be passing this out. And in the email, I will include Andrea's information that she included for the free 15-minute kind of clarity call with Genomicare. So for people who have more specific questions or want to know if genetic counseling is right for them or have a few more questions around that, then we will get that information out to you as well for sure. Um, is there anything else specifically in the chat box here, ladies, that you think we haven't touched on with it specific to genetic counseling, PGT-A, anything like that?
I noticed a couple questions about timing of carrier screening. It's not too late. It's not too late to do it if you're already in a cycle, or if you've already, sent your embryos off for PGT-A. Um, we just would prepare you for like the chance that you are that 2 to 3%, you are both carriers for the same thing. That might add time later downstream, and a little bit of headache to you. But the 97% of the time, 98% of the time that you're all clear, then yeah, it's honestly never too late to do PGT to do carrier screening.
And actually, one question that often comes up that I did see in the chat is regarding the blastocyst and the biopsy. So, Dr. Cantor, can you talk a little bit about, someone is asking if there's a chance of, if there's risk to the blast from the biopsy?
Yeah, so the biopsy that the embryologists are taking is really of very few cells of the outer layer of the embryo. So the embryo, as Andrea mentioned, it's made up of different parts. There's that inner cell mass that ultimately becomes the baby, and there's that outside layer of the embryo that ultimately will become the placenta. The biopsy that the embryologist is taking is of very few cells. It's about 5 to 10 cells, and it's from that outer layer of the embryo. It's not of the portion of the embryo that's ultimately going to become baby. So risk of, causing harm to the embryo or embryo damage is very low. Typically I would say, even like less than 2%. If an embryo for whatever reason is not going to has an issue at time of biopsy or doesn't survive a biopsy, which is truly very uncommon because we're only biopsy embryos that have reached a specific developmental stage, then I would say that embryo probably wasn't an embryo that was, going to result in a baby, unfortunately. So we can't just biopsy any embryo. We have specific, the embryo has to make it to a specific stage to be able to be biopsied. And typically the biopsy is done of that outer layer that we assume is representing the embryo in full. So ultimately, risk to damaging that embryo is quite low, ultimately.
Great, thank you. I want to be respectful here of everyone's time. I know there are a few more questions, and again, we will get to you an embryo grading webinar. And then I noticed there were a few questions in there about influencing the entire IVF cycle as a whole and understanding it, and also influencing our egg and sperm health. So in the email I sent tomorrow, we have done some webinars on that, and we do have some great resources online, webinars like this that we have posted as videos. So I will share where everyone who's on this call can find that information. And thank you very much, everyone, for joining us tonight. Thank you to both of you for being such wonderful speakers.